Welcome to the PubMed Daily Review for May 27, 2026. Today we scanned 592 new open access papers across 133 journals. In this video, we'll cover 10 trending research topics and 10 selected papers for detailed analysis. Trend 1 of 10, AI for image analysis and sensing. AI models are applied to analyze and interpret various types of images for sensing, detection, and understanding tasks across different domains. This cluster covers 11 papers. Representative work includes TDDC-YOLO, texture-defect disentanglement for robust wood surface crack detection under domain shifts, predicting fire consequences with a transformer model based on multimodal feature fusion, and optimization of image restoration technology and AI iterative upgrade based on PCNN. Trend 2 of 10, clinical studies, neurological and organ health. Clinical research on neurological disorders and their impact on organ health, function, and multimorbidity. This cluster covers 66 papers. Representative work includes Prise in charge de la multimorbidité, Cerveau, un guide de pratique clinique, Stereopsis deficits in schizophrenia, a systematic review with exploratory meta-analysis, and impact and management of cognition on speech and swallow function in Parkinson's disease. Trend 3 of 10, diverse fundamental scientific research. This trend encompasses foundational investigations across a wide array of scientific disciplines, pushing the boundaries of knowledge. This cluster covers 51 papers. Representative work includes defect-mediated N-type to P-type transition and enhanced thermoelectric performance in by 1.8x, Ni, SB, 0.2, Te3, alloys, the limits of language, and rational design of ligand-free Araceous nanotrees for plasmonic tip localization and advanced third-order nonlinear optics. Trend 4 of 10, cancer research, mechanisms, and therapies. Exploring diverse cancer mechanisms, novel therapeutic strategies, and diagnostic advancements to combat oncological diseases. This cluster covers 61 papers. Representative work includes efficient and metal catalyst-free approach towards highly substituted N-sulfonylpiperidine-mono, Spiro, 1, 2, 4, 5, tetraoxanes for potential applications in chemotherapy, hallmarks of epithelial mesenchymal plasticity in cancer, and cracking the shield. Oncolytic viruses versus the tumor immune fortress. Trend 5 of 10, environmental modeling and resource management. Utilizing diverse modeling and analytical techniques for environmental assessment and sustainable resource management across various domains. This cluster covers 77 papers. Representative work includes synthesis, in vitro and in silico bioactivity, and computational studies of pyrene and p-methoxycinnamaldehyde-linked 4-hydroxycoumarins, risk prevention and control evaluation and optimization strategy for digital transformation in oil and gas production management areas, and the benefit of advanced mapping techniques to address the spatial distribution of soil sulfur total, gypsum-based, and total reduced in the dried Sawa lakebed. Trend 6 of 10, agricultural genetics and animal science. Investigating genetic, biological, and omics factors in agricultural animals and plants to enhance production and quality. This cluster covers 50 papers. Representative work includes effects of genotype and sex on carcass characteristics, meat quality, volatile compounds, and flavor profiles of Thai native chickens and broilers, multi-omics-based characterization and differentiation of fecal microbiota and metabolite profiles in high and low-yielding laying hens, and genetic diversity and population structure of inset, in-seat ventricosum, well. Cheeseman, landraces from Gidio and Wolata zones, and Sidama regional state, southern Ethiopia using SSR markers. Trend 7 of 10, clinical trials and medical interventions. Research evaluating medical treatments, patient care strategies, disease characteristics, and health information tools through various study designs. This cluster covers 60 papers. Representative work includes immediate implant placement in type 2 socket using vestibular socket therapy with pericardial membrane versus simultaneous guided bone regeneration, randomized controlled clinical trial, opioid-free versus opioid-based anesthesia in laparoscopic bariatric surgery, an exploratory randomized pilot trial, and prevalence and determinants of secondary hyperparathyroidism, IMWES, 2021. Trend 8 of 10, infectious diseases and antimicrobial resistance. This trend explores infectious diseases, including their diagnosis, management, and the growing challenge of antimicrobial resistance. This cluster covers 60 papers. Representative work includes genomic and phenotypic characterization of carbapenem-resistant Acinetobacter baumannii isolates from patients and trolleys and their potential connections in an intensive care unit, association between thrombocytopenia and Plasmodium falciparum infection in Ghana, and diagnosis, management, and clinical outcomes of childhood invasive aspergillosis. Trend 9 of 10, Public Health and Healthcare Access, examines healthcare access, public health systems, and the impact of health services on diverse populations and specific medical conditions. This cluster covers 79 papers. Representative work includes effects of multi-channel consultation on e-healthcare demand, an empirical study, un système de santé privé parallèle in faire que déteriorer l'hs aux interventions chirurgicales finances par l'État, and le lichen plan palmoplantaire. Trend 10 of 10, AI and machine learning applications. Applying AI and machine learning techniques across diverse domains for problem-solving and analysis. This cluster covers 77 papers. Representative work includes a multi-domain graph-integrated neural framework for robust acoustic anomaly detection under adverse environmental conditions, microRNAs in endometriosis, bioinformatics resources, machine learning strategies, and multi-omics perspectives, and a self-supervised GNN transformer framework for weak microseismic signal identification. Up next, our first paper out of 10, Interferometric Scattering Microscopy Supports Real-Time Mass-Resolved Label-Free Single-Molecule Immunoassay by Karasi Brower and Edward Grant from Department of Chemistry, University of British Columbia, Vancouver, British Columbia, V6T1Z1, Canada. This paper was selected because it introduces a groundbreaking label-free, real-time, mass-resolved single-molecule immunoassay, which represents a fundamental technological leap in studying protein interactions. Let's break down its key contributions. Understanding how proteins interact is crucial for deciphering biological processes and developing new therapies. However, our current methods often rely on indirect measurements or chemical labels, which can hide the true dynamics of these interactions. This limitation has hindered our progress in understanding fundamental life processes and discovering new treatments. To overcome these challenges, researchers developed a novel bioaffinity iSCAP platform. This technique uses interferometric scattering microscopy to directly observe individual protein binding events without the need for labels. By detecting light scattered from single proteins as they bind to an antibody-functionalized surface, the platform achieved single-molecule sensitivity and could even discriminate by molecular weight. They tested this innovative approach using IgM as a model system and in human serum. The results were quite impressive. The measured association rates scaled linearly with concentration over an impressive 3 orders of magnitude, demonstrating the technique's quantitative power. Crucially, direct counts of IgM binding events in human serum yielded concentrations that agreed well with bulk measurements obtained by traditional ELISA. Furthermore, the platform successfully captured the elementary dissociation rate constant, K-off, from the distribution of single-molecule dwell times, showcasing its ability to resolve dynamic processes at the molecular level. This groundbreaking work introduces a powerful new tool for biomedical research. The bioaffinity iSCAP platform offers a general framework for quantitative, single-molecule immunoassays, providing unprecedented insights into protein interactions. While its potential is vast, future studies will need to explore its application to a wider range of biomolecules and more complex biological samples. Ultimately, this label-free, real-time approach holds immense promise for advancing our understanding of disease mechanisms and accelerating the discovery of new therapeutics. Up next, our second paper out of 10, Explainable Machine Learning-Guided Integrated Multiomics Analysis reveals macrophage-driven immune suppression in breast cancer by Yunus Azamzad and Alvaro Conluque from Oslo Center for Biostatistics and Epidemiology, University of Oslo, Oslo, Norway. This paper was selected because it leverages cutting-edge explainable machine learning and integrated multiomics analysis to uncover critical macrophage-driven immune suppression mechanisms in breast cancer, significantly advancing our understanding of the tumor micro. Let's break down its key contributions. Understanding the complex cellular landscape within the tumor microenvironment, or TME, is crucial in breast cancer. Despite extensive research, we still don't fully grasp how these cellular compositions impact disease progression and patient prognosis. This knowledge gap highlights a critical need to identify new targets that could significantly improve treatment outcomes for patients. Researchers employed an explainable machine learning pipeline to analyze cellular fractions within the tumor microenvironment. They scrutinized data from over 5,000 patient samples across the metabric and TCGA datasets. The study compared associations with relapse-free survival and pathological complete response, focusing on specific breast cancer subtypes. This comprehensive approach aimed to uncover the complex roles of TME cells in disease progression. The study uncovered a fascinating dichotomy. Macrophages correlated positively with pathological complete response, but negatively with relapse-free survival. This was particularly evident in ER+ and luminal A/IB breast cancer subtypes. Further analysis showed these macrophages accumulated near HLA-B epithelial cells and were strongly associated with regulatory and exhausted T cells, suggesting an immunosuppressive role. Key molecular interactions involving ligands like SIGLEC9 and ALCM were also predicted. This research significantly enhances our understanding of macrophage-associated immune suppression in breast cancer, particularly in ER+ and luminal A/IB subtypes. The identification of HLA-B macrophages and their specific interactions with TREG and TEC cells offers crucial insights into the mechanisms of immune evasion. Critically, these findings pinpoint novel molecular targets like SIGLEC9 and ALCM, paving the way for the development of innovative immunomodulatory strategies to improve patient outcomes. Up next, our third paper out of 10, Understanding Ukraine's Immunization Supply Chain Resilience in the Face of War. A mixed-method study by Stanislav Gajewski and Yarno Habit from World Health Organization Country Office in Ukraine, 9b, Khrushchevskoho Street, Kyiv, Ukraine. This paper was selected because it addresses an exceptionally pressing and unique global health challenge by providing critical insights into maintaining immunization supply chain resilience in an active war zone, offering invaluable lessons for humanitarian aid and global. Let's break down its key contributions. The ongoing conflict in Ukraine has placed immense strain on critical infrastructure, including the nation's immunization supply chain. Despite these challenges, vaccine availability has largely been maintained. This study sought to understand how the Ukrainian ISC adapted to these unprecedented disruptions. It's crucial to learn from these experiences to build more resilient healthcare systems in conflict zones globally. Researchers employed a mixed methods approach to thoroughly investigate the ISC's adaptation. They conducted 15 in-depth interviews with key stakeholders across various levels, from national to local facilities. This was complemented by a comprehensive review of literature, regulatory documents, and program data. The analysis used a health system resilience framework to interpret the findings, providing a robust understanding of the challenges and successes. The study revealed several key enablers of resilience. Digitalization of stock management and extensive training significantly strengthened the ISC's knowledge capacity. Crucially, large investments in WHO pre-qualified cold chain equipment and backup power solutions dramatically improved the system's ability to manage electricity outages. While governance and coordination were enhanced through centralization and collaboration, persistent barriers included gaps in population data for accurate forecasting. Overall, the ISC continued to function despite repeated attacks. This study offers vital insights into maintaining essential health services during conflict. The findings underscore the importance of proactive investments in digital infrastructure and cold chain capacity for supply chain resilience. While Ukraine's ISC showed remarkable adaptability, addressing data gaps and reducing donor dependency are crucial next steps. Ultimately, these transferable lessons can inform strategies for strengthening immunization programs and global health security in other vulnerable settings worldwide. Up next, our fourth paper out of 10, IC's Atlas reveals a suppressive NK cell niche in pan-cancer immunotherapy profiles by Yu Ma Xie and Huichuan Yu from Department of General Surgery, Colorectal Surgery, the 6th affiliated hospital, Sun Yat-sen University, Guangzhou, China. This paper was selected because it addresses the critical challenge of immune checkpoint inhibitor resistance by developing a comprehensive pan-cancer dataset and identifying a novel suppressive NK cell niche, offering significant implications for improving cancer immunotherapy outcomes. Let's break down its key contributions. Immune checkpoint inhibitors, or ICIs, have truly transformed cancer therapy, offering new hope for many patients. However, we've observed that a substantial number of individuals don't respond to these treatments, highlighting a critical gap in our understanding. This lack of response is a major challenge, and it's partly due to the absence of comprehensive, harmonized data and tools to systematically uncover why some patients benefit while others don't. This paper tackles that very problem head-on. To address these challenges, the researchers first developed a powerful new resource called IC's Atlas. This comprehensive platform integrates curated transcriptomic and clinical data from over 1,000 ICI-treated patients across 8 different tumor types. They then applied this ICIS Atlas framework to conduct a systematic pan-cancer analysis, comparing responders to non-responders. This approach allowed them to delve deep into the molecular landscape to uncover critical differences. Using the ICIS Atlas, the team made some exciting discoveries. They identified universal signatures that were specific for ICI response, including beneficial interactions among immune cells. Crucially, they uncovered a competitive cell community and, more specifically, a SERPING1 plus VEGFA plus natural killer cell-mediated immunosuppressive niche present in patients who didn't respond to treatment. These findings were further validated using advanced techniques like single-cell and multiplex immunohistochemistry, strengthening the evidence for this novel mechanism. This study is incredibly significant because it not only provides a powerful new tool, the ICIS Atlas, but also uncovers a critical mechanism of resistance to immune checkpoint inhibitors. The discovery of the SERPING1 plus VEGFA plus NK cell-mediated immunosuppressive niche offers a concrete, novel target for developing new therapies to help non-responders. Moving forward, this resource and these findings will be instrumental in guiding future research, paving the way for next-generation biomarkers and more effective treatment strategies in cancer immunotherapy. Up next, our 5th paper out of 10, Tumor Treating Field plus Chemotherapy for Locally Advanced Pancreatic Adenocarcinoma, an international cost-effectiveness analysis by Kuan Liu and Hong Zhu from Department of Radiation Oncology, Xianya Hospital, Central South University, Changsha, Hunan, China. This paper was selected because it addresses a critical and pressing challenge in oncology by providing a comprehensive cost-effectiveness analysis of a novel therapy for pancreatic adenocarcinoma, which is essential for guiding clinical implementation and healthcare policy decisions. Let's break down its key contributions. The PANOVA-3 trial recently demonstrated that adding tumor-treating fields, or TT fields, to standard chemotherapy significantly improved survival for patients with locally advanced pancreatic adenocarcinoma. But with the high cost of these innovative therapies, it's crucial to understand their economic impact. This study aimed to evaluate if this survival benefit translates into a cost-effective treatment strategy. Researchers developed a 3-state Markov model, utilizing clinical data from the PANOVA-3 trial to simulate patient outcomes. They compared the cost-effectiveness of TT fields combined with chemotherapy against chemotherapy alone. The analysis considered both American and Chinese healthcare payer perspectives, with specific willingness to pay thresholds for each region. The analysis revealed that adding TT fields resulted in incremental costs of over $147,000 in the USA and nearly $40,000 in China, yielding modest KELI gains. This translated to incremental cost-effectiveness ratios of over $1 million per KELI in the USA and nearly $280,000 per KELI in China. Both figures significantly exceeded their respective willingness to pay thresholds, indicating that at current prices, TT fields plus chemotherapy was not a cost-effective strategy. This study highlights that while TT fields offer clinical benefits for locally advanced pancreatic adenocarcinoma, their current price makes them not cost-effective from a healthcare payer's perspective in both the US and China. The device cost was identified as the most significant factor influencing this outcome. Moving forward, exploring price adjustments or innovative funding models will be crucial to make this promising therapy more accessible and economically viable for patients. Up next, our 6th paper out of 10, towards simultaneous pseudospace reconstruction and cell-type deconvolution of single-cell spatial transcriptome using SPA-Dicer by Junming Zhang and Hui Lu from College of Computer and Information Engineering, Nanjing Tech University, Nanjing, Jiangsu, China. This paper was selected because it introduces a highly novel deep learning framework, SPA-Dicer, that addresses a critical and pressing challenge in integrating single-cell RNA sequencing and spatial transcriptomics, thereby significantly advancing our ability to. Let's break down its key contributions. Understanding tissue heterogeneity is crucial in biomedical research. We have powerful tools like single-cell RNA sequencing and spatial transcriptomics, but each has its limitations. scRNA-seq gives us high resolution but no spatial context, while spatial transcriptomics provides location but at lower resolution. This paper addresses the challenge of bridging these gaps to gain a more complete picture. The researchers developed SPA-Dicer, a novel deep learning framework. This method was designed to simultaneously reconstruct the pseudo-spatial localization of single cells and deconvolve cell types within spatial transcriptomic spots. Key to its design were cross-domain feature disentanglement, and an adaptive loss weighting strategy. They rigorously evaluated SPA-Dicer's performance on both simulated and real-world human tissue datasets. The results were very promising. SPA-Dicer demonstrated consistent improvement over existing state-of-the-art methods. Specifically, it showed enhanced accuracy in both reconstructing cellular spatial localization and resolving cell type composition. This superior performance was observed across all 7 datasets tested, including various human tissue contexts, highlighting its robustness and broad applicability. This study introduces SPA-Dicer as a powerful new tool for spatially informed biological discovery. By unifying pseudospatial reconstruction and cell type deconvolution, it overcomes significant limitations in current single-cell and spatial transcriptomics technologies. Looking ahead, SPA-Dicer could be applied to complex disease contexts or integrated with other omics data to unlock deeper insights into tissue organization and disease mechanisms. Up next, our 7th paper out of 10, using deep learning to identify brain networks mediating cognitive and motor impairments in alcohol use disorder by Eason Wang and Killian M. Pohl from Department of Bioengineering, Stanford University, Stanford, California, USA. This paper was selected as the top choice because it leverages a sophisticated deep learning framework to uncover fundamental brain network mechanisms underlying alcohol use disorder, a major public health challenge, representing a significant advancement in understanding. Let's break down its key contributions. Alcohol use disorder, or AUD, affects a significant portion of the population, leading to various cognitive and motor impairments. These impairments can severely impact daily life and overall well-being. This study aimed to explore the underlying brain mechanisms responsible for these deficits, using advanced analytical techniques to differentiate individuals with AUD from healthy controls. Researchers utilized a data-driven deep learning framework to analyze complex brain data. They studied human participants with AUD and controls, examining resting state fMRI connections and a battery of neuropsychological tests. The goal was to identify specific brain networks and their underlying mechanisms that contribute to AUD-related impairments. The study revealed crucial insights into the brain networks involved in AUD. They found that the temporal attention network, or TN, fully mediated the impact of AUD on several key functions, including spatial working memory and attention. Additionally, the sensoromotor network played a role in motor performance. These findings were robust, even replicating in an independent cohort of individuals with HIV, reinforcing the identified brain behavior relationships. This research significantly advances our understanding of the neurobiological underpinnings of alcohol use disorder. By pinpointing specific brain networks, we now have potential targets for future interventions and therapies. While these findings are promising, more work is needed to translate this mechanistic knowledge into practical clinical tools that can improve the lives of individuals with AUD. Up next, our 8th paper out of 10, durvalumab plus enlotinib versus durvalumab alone as maintenance treatment in extensive-stage small cell lung cancer. Durable, a multi-center, randomized, Phase 2 trial and biomarker analysis by Bo Zhang and Hua Zhong from Department of Respiratory and Critical Care Medicine, Shanghai Chest Hospital, Shanghai Jiao Tong University School of Medicine, Shanghai, China. This paper was selected because it presents a multi-center, randomized, Phase 2 clinical trial demonstrating a novel maintenance therapy combination for extensive-stage small cell lung cancer, directly addressing. Let's break down its key contributions. Despite recent advancements in treating extensive-stage small cell lung cancer, or ESSCLC, with first-line immunotherapy and chemotherapy, the survival benefits for patients are still quite limited. This highlights a significant unmet need for more effective treatments to maintain remission and extend lives. This research paper addresses this gap by investigating whether adding a new agent, enlotinib, to standard durvalumab maintenance therapy could improve patient outcomes. It's a crucial question for improving care in this aggressive cancer. The Durable study was a prospective, multi-center, open-label, randomized, Phase 2 trial. It enrolled 66 patients diagnosed with extensive-stage small cell lung cancer who had completed first-line chemotherapy. Patients were then randomly assigned to receive either durvalumab alone or a combination of durvalumab and enlotinib as maintenance therapy. The primary goal was to evaluate progression-free survival, assessed by an independent central review, to ensure unbiased results. The results were quite promising. The combination therapy of durvalumab plus enlotinib significantly improved progression-free survival, with patients experiencing a median PFS of 5.4 months. This was a substantial increase compared to just 1.9 months for those on durvalumab alone. The hazard ratio of 0.64 indicated a 36% reduction in the risk of progression or death with the combination. While the p-value was 0.12, it met the study's predefined significance threshold of less than 0.20. However, it's important to note that the combination group did see a higher incidence of grade 3 to 4 treatment-related adverse events, at 24.2%. This study suggests that the combination of durvalumab and anlotinib could be a valuable new maintenance treatment option for patients with extensive-stage small cell lung cancer, offering a significant improvement in progression-free survival. However, clinicians must carefully weigh the benefits against the increased risk of grade 3 to 4 adverse events associated with the combination. Moving forward, larger confirmatory trials are essential, and further investigation into biomarkers could help us identify which patients are most likely to benefit from this combination, paving the way for more personalized treatment approaches. Up next, our 9th paper out of 10, Mitochondrial Flagella-like Extensions, MitoFLARE, Dysfunction Triggers Sting-Mediated Immune Dysregulation in Sepsis by Weilong Hong and Qinyang Duan from Department of Critical Care Medicine, the second affiliated hospital of Chongqing Medical University, Chongqing, China. This paper was selected because it uncovers a novel and fundamental mechanism involving mitochondrial dysfunction and sting-mediated immune dysregulation in sepsis, a critical and widespread clinical challenge with high mortality. Let's break down its key contributions. Sepsis is a critical condition where the body's immune response to infection goes awry, leading to widespread damage. We know that mitochondria are vital for cellular function, but their role in the immune dysregulation seen in sepsis has been less clear. This research aimed to uncover how mitochondrial communication and dysfunction contribute to the progression of this severe disease. To explore these complex mechanisms, researchers utilized an in vitro model, treating cells with LPS to mimic early sepsis. They meticulously examined mitochondrial remodeling, including the formation of unique flagella-like extensions called MitoFLARE. The study then tracked how these structures changed as inflammation progressed, ultimately linking their dysfunction to the release of mitochondrial DNA and activation of critical immune pathways. The study revealed fascinating dynamics. Initially, mitochondria formed protective MitoFLARE structures, driven by specific protein interactions, to maintain internal communication. However, as inflammation worsened, these MitoFLARE functions were suppressed due to disruptions in key mitochondrial complexes. This suppression ultimately led to the rupture of the outer mitochondrial membrane, releasing mitochondrial DNA into the cytoplasm. This released DNA then triggered the powerful C-gas sting pathway, escalating the immune response and contributing to the severe inflammation seen in sepsis. This research offers crucial insights into the pathogenesis of sepsis, revealing how mitochondrial dysfunction directly fuels the immune dysregulation. Understanding the intricate dance of MitoFLARE and mtDNA release opens up exciting new avenues for therapeutic intervention. While these findings are foundational, they lay a strong theoretical basis for developing innovative treatments that could one day mitigate the devastating effects of sepsis. Up next, our 10th paper out of 10, long-read deep sequencing reveals high rates of multilineage transmission and rapid viral population changes in acute HIV infection by James I. Mullins and Carolyn Williamson from Department of Microbiology, University of Washington, Seattle, Washington, USA. This paper significantly advances the state-of-the-art by revealing a surprisingly high rate of multilineage HIV transmission and rapid viral population changes in acute infection, fundamentally altering our understanding of early HIV biology with. Let's break down its key contributions. Hello, everyone. Today, we're diving into a fascinating study that explores the very beginning of HIV infection. Understanding how the virus establishes itself and changes in these crucial early moments is absolutely vital for developing new ways to prevent HIV and for designing more effective vaccines. This paper really helps us fill in some of those critical knowledge gaps. The researchers utilized data from the FRESH and RV217 cohorts, along with participants from the AMP trials, to get a very precise look at early HIV infection. They employed long-read deep sequencing, a powerful technique, to analyze samples taken within a median of just 4 days between a negative and a positive HIV test. This allowed them to capture an incredibly early and detailed snapshot of the virus's initial stages. The findings were quite striking. They discovered that 37% of infections involved multiple transmitted viruses, which is much higher than previously thought. Furthermore, in 2/3 of these multilineage cases, the relative abundance of different viral lineages fluctuated substantially over time. Despite these changes, the overall viral populations showed limited diversity and evidence of selection on important proteins like GAG and ENV, with negative selection being particularly evident. This research has significant implications. The surprisingly high rate of multilineage HIV transmission means we might need to rethink how we approach prevention and design future vaccines. The rapid and substantial changes in viral populations early on also underscore the incredible adaptability of HIV, which is crucial for understanding its long-term persistence. Ultimately, these findings offer exciting new targets for intervention and help explain some of the challenges we've faced in developing effective HIV vaccines. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.